In first step, cell injury, we have seen the chart. In second video, we seen the mitochondrial injury. In third, we are seeing the loss of calcium homeostasis. If there will be increase influx and release of calcium from intracellular store. Where at the intracellular store, it is on mitochondria and endoplasmic reticulum. Mitochondria and endoplasmic reticulum contains calcium inside them, but during injury there is increased influx of calcium that lead to increased cytosolic calcium level. Increased cytosolic calcium level activate phospholipase, protease, endonuclease and ATPase. These are enzymes that get activated when there is increased level of calcium inside the cytoplasm of the cell, where generally occurs in the mitochondria, where phospholipase and protease get activated, and phospholipase cause membrane damage and protease cause breakdown of proteins, intracellular proteins, cytoskeletal proteins. If endonuclease get activated, it break down the DNA. There will be DNA fragmentation that leads to nuclear damage, and if ATPase get activated, it decreases the ATPase, it breaks down the ATP. That's why there is decrease of ATP. The next mechanism of cell injury is free radical. First one is hypoxia or ischemia, the most common cause. Second is mitochondrial injury. Third is increased cytosolic calcium level or the loss of calcium homeostasis, and the fourth one is free radical. Free radical are normally ATP is generated by oxidative phosphorylation. This oxygen is converted into O2 minus, that is superoxide, that will convert into H2O2, that is peroxide, that will convert into hydroxyl ion OH minus ion, that contains most number of electrons. It's more danger. The number of free electron increases, that will increases the reactivity of that species, and there will be conversion of this hydroxyl ion into water by radiolysis. It is an interconversible step, means it can be converted into water or water can be converted into hydroxyl ion. Generally it is generated within the mitochondrial inner membrane. Mitochondria has inner membrane, outer membrane. In the inner membrane it is generated within the inner membrane. Effect is, if the free radical gets outside the inner membrane or get increased inside the inner membrane, that will lead to damage of the protein, lipid, nucleic acid and carbohydrate. There will be lipid peroxidation of cell membrane. Lipid peroxidation will lead to DNA damage, that will lead to membrane damage. Membrane damage leads to liberation of intracellular enzyme. See, lipid peroxidation, DNA damage leads to membrane damage. Membrane damage leads to liberation of intracellular enzyme. There will be nuclear, cellular and serum enzyme estimation. Now the third one is protein cross linkage will be there, and fourth one is oxidative or oxidation of amino acid and protein that result in fragmentation. How does this free radical now increase free radical? How will remove the free radical? If there is increase of free radical level, we will remove them, we will not remove. There are two things like scavenger for free radical. They are known as scavenger for free radical, means they eat the free radical. How they eat? It means they convert it. Superoxide dimutase, they are present in mitochondria and cytosol. They convert the superoxide into hydrogen peroxide. Superoxide dimutase is a scavenger of free radical. There are also other scavenger of free radical like glutathione peroxide, which convert the superoxide into peroxide. It is present in mitochondria and cytosol. Third scavenger is catalase, that is present in peroxisome. Catalase enzyme is present in peroxisome that converts peroxide into water. Vitamin E, vitamin C, transferrin, ferritin, ceruloplasmin are also scavenger of free radical. Now this completes the first question, cellular adaptation and mechanism of cell injury. Thank you.